All right, folks, we're going to go ahead and get started. Thank you all for coming to the, what month is it? April. No, sorry, May. May RIDE seminar. Today we have two internal speakers, internal to the CVM. We have Dr. Noel Noyes and Dr. Steve Friedenberg. And I'm just here to welcome you all to the RIDE seminar series, and I'm going to hand it over to one of the folks in Noel's lab to introduce her. I think the folks in the research office had a great idea to have the trainees or colleagues of the speakers introduce them. And I thought this was a great idea because it helps everybody know everybody else and who they're working with. So that's what we're starting here this month. So I'm going to hand it over.
Thank you. Good afternoon. Thank you all for joining us today. My name is Tara Gear. I am a postdoc at Department of Veterinary Population Medicine. And it's my great pleasure to introduce my supervisor and mentor, Dr. Noel Noyes, who will be speaking at today's RISE seminar. As many of you already know, Dr. Noyes is an associate professor at BPM and also McKnight Land Grant Professor at University of Minnesota. She joined BPM in early 2018 after completing PhD and DVM from Colorado State University. So in addition to DVM and PhD, she also received a BA, Bachelor of Arts degree in European Study from Amherst College, Massachusetts, and EMA from Germany as a part of independent research, as a part of the German Transfer Fellowship called Alexander von Humboldt Fellowship. So she has a quite diverse academic background. And our research primarily focuses on epidemiology of antimicrobial resistance, microbial genomics, and I have personally learned so much from her. And today's Dr. Noyes will be sharing this interesting idea of pooling in the context of microbiome research, whether pool sample can actually use in the context of the large field-based microbiome study. So without further delay, let us welcome Dr. Noel Noyes.
Thank you. I have this one. Thank you, Tara. So that was the perfect person to introduce me because Tara really was the driver of this pilot study that I'm going to present today that we're still working on. So it's sort of in progress. And Tara did the sampling, a lot of the study design, the analysis that I'm going to present today. So thanks, Tara, for that. This pilot study is part of a larger grant from the USDA, and co-PIs on that grant are Drs. Singer and Peters. And we're also doing that in collaboration with Mark Schwartz, who's faculty here in VPM and also owner of Schwartz Farms, where we conducted this pilot study. So thank you to all of the people who've been involved so far. So to give some background, first, let's just say, what am I talking about when I say a pooled sample? I gave the wrong version. Okay, well, I'm gonna wing it. So a pooled sample, there's like so much terminology around here that gets thrown around in pooled composite samples. I'm just gonna keep it simple and say for now, a pooled sample is a single sample that represents a group of individuals. So rather than one sample equals one individual, now we have one sample equals many individuals. And in livestock research, there's a long history of using pooled samples for, both research and diagnostic purposes. And this was actually an email that hit my inbox yesterday at 5:20, and it just proves my point that, this is both has a long history and is also still very relevant today. This is National Hog Farmer talking about use of pooled sampling strategies for influenza detection in pigs. Most of the previous research, I would argue like 99.9% of it, focuses on the use of pooled samples for detecting specific microbes. And usually the questions are around sensitivity and specificity of being able to detect these things accurately within these pooled samples. There's actually very little research about use of pooled samples for microbiome studies. I did a quick search a while ago, probably about a year ago now, and I found two papers that specifically address this question of pooled samples for microbiome research. One of them is in the human literature, and the other one is actually in swine. So In addition to this large gap about pooled samples, we also are seeing an increase in the number of large population-based studies in livestock research that are asking microbiome type questions. And that's because we realize we need better powered studies within microbiome research generally. And so you have these large, very large studies in livestock, and they generate a lot of samples. And it's sort of common sense, I think, to ask the question, could we pool some of these samples to be more efficient with our resources? And that's actually the sort of very practical question that motivated this pilot study, because we were launching this larger USDA grant where we were going to collect about above 12,000 individual samples from pigs. And one of the questions was, could we save costs by pooling? So that's the first very obvious benefit of pooling, right? Is that you can save costs on sample assays. The other benefit that actually may not be as apparent, but I think is a really interesting one to ask about, is the unit of interest of the research question. And in livestock research, it's often groups of animals. It's not like your individual cat or dog, but it's like a litter of pigs. And the intervention and the treatment is applied at the level of the litter or the group of animals, not to individuals. And so when we're looking at the effect of the treatment or the intervention, we're actually wondering about the effect across the population or the group. So in that way, maybe pooled samples are actually a better reflection and more cost effective. And then some people hypothesize that pooling samples may increase our sensitivity to detect rare microbes that might be circulating within the population. I actually don't subscribe to that hypothesis, but it's an interesting one that hasn't been fully answered. So there are several important open questions. This is just 3 of them listed here, and I'm only going to focus on the first one in this talk because it's 20 minutes. The first one is that when you think about specific microbes, it's pretty clear, I want to know sensitivity, specificity. When you think about the microbiome, we're detecting like thousands of different microbes, and we analyze the data in a different way usually, where we're more interested in like overall diversity of the microbiome, and overall composition of the microbiome. And we don't know how pooling impacts those metrics because they are not relevant for detecting specific pathogens, for example. There's other things that are very interesting to ask about the role of pooling, like how does it interplay with variability between individuals within a population, and how does it interact with sequencing depth, which is an important parameter for sort of sensitivity of a metagenomic or a microbiome assay. I'm not going to talk about those today, but this pilot does help us answer some of those questions as well. Okay, so overall goal of this pilot was really to understand, do composite samples equal pooled samples equal individual samples? Now I'm going to get more specific on these definitions in the context of this pilot. Individual samples, one sample represents one individual. Pooled samples, we collect the samples from individuals and then pool them. For composite samples, we actually sample the environment of a group of animals, and that is representing the group, okay? So those are the definitions. And again, we want to understand if there's differences in these different workflows in terms of microbiome diversity and composition. So the methods for this pilot, we went into a sow farm on a single day, and we enrolled 10 litters of day-old piglets and 10 litters of 20-day-old piglets. So the sow IDs on the bottom there represent the 20 litters. And we picked these days of age because we know that the microbiome shifts very dramatically in the pre-weaning period. So both of these age groups, the pigs are still with the sow in a pen. And we wanted to know if we saw the same patterns in terms of pooling in the young pigs and then the just pre-weaning pigs. So we went in, and of course, every sow has a variable number of piglets. So the smallest piglet sizes had 8 piglets, and then the largest litters had 16 piglets, right? But no matter the litter size, we sampled all the piglets, and we took individual rectal fecal swabs from each piglet. So we had 258 individual fecal swabs that we sampled on this one day, representing 20 litters. And we also, in each physical pen in the barn, we took a swab and we swabbed like 3 areas of the pen that had fecal matter, and we did that 2 more times for each pen. So we had 3 composite pen floor samples, I'll call them, for each litter of pigs. So that was 60 composite pen floor samples. So we brought these raw swabs back to the lab, and I'm going to tell you how we processed them for pooling. So we took the individual fecal samples and processed them, basically made a slurry, and then aliquoted that. And one aliquot stayed as an individual sample, and the other aliquot got pooled so that we had 4 pools per litter of pig. And so if you have a litter with 8 piglets, then each pool has 2 piglets in it, right? If you have a litter of 16, each pool has 16 or has 4 piglets in it. So we kept the number of pools per litter consistent, but the number of pigs per pool would be different based on the litter size. And so we had the raw material there, and then we extracted DNA from both the individual aliquots and also the fecal pool aliquots. Then we took the DNA from the individual samples over here in blue, and we pooled the DNA. And we used the exact same strategy as we did for the fecal pool. So now we have 4 DNA pools per litter for a total of 80. And then from each of these 3 workflows and those libraries, we did library preparation where we targeted the V4 region of the 16S gene, which is a common target for microbiome analysis. And then for the composite pen floor samples, it was a much simpler workflow. We just took those 60 samples, extracted the DNA, did library prep. So these are our 4 sort of treatment groups, or I'm going to call them workflows: individual DNA pools, fecal pools, composite floor pools. We then randomized those libraries into plates, and we sequenced them, and we performed bioinformatic and statistical analysis. So the results. So the first question that we have is, what about the overall composition of the microbiome? There's like thousands of bacteria in these samples, and we want to know, broadly speaking, does the profile of the microbes look the same based on workflow, or does it look different? The way that we do that is we do a beta diversity analysis, and we compare the overall profile using like a data reduction technique, and we use ordination to visualize those results. So this is an ordination plot where each dot on the plot represents a single library, and we've colored the dots, and we've also used in different shapes of symbols. So the triangles, oh, and I should also say, the closer the dots are together in this 2D space, the more similar their microbiome profile is, and dots that are further apart have a less similar microbiome profile. So we look for patterns of clustering. And what you can see is that all of the triangles are on the left-hand side of this ordination plot, and all of the circles are on the right-hand side, and those are the samples from the 20-day-old and the 1-day-old pigs, respectively. So there's clear separation of the microbiome of the day 1 and the day 20-day-old pigs, which is completely expected. That's why we sampled both groups, because we knew their microbiomes would be very different, right? So that's good. We confirm our expectations going in. The next thing I want to point out are the 4 colors. Those are the 4 workflows that I just showed you on the previous slide, right here, right? So the ellipses indicate like the 95, let's say a 95% confidence interval around like the average microbiome profile for each workflow. And if you sort of use your imagination and put like a center dot within each ellipse, that would be kind of like the average microbiome profile for that set of samples. And you can see that the composite floor samples in black are just very different than the other 3 workflows. So that was our first sort of indication that we probably can't use pen floor samples to represent the litters of pigs, because they have a different profile. So then I want to call your attention to the other 3 circles, the red, yellow, and blue. Those are from the individual and the fecal and DNA pools. The first thing I want you to notice is that they're sort of concentric, right? They're like stacked within each other. And if you imagine that center point, they're lined up on top of each other, right? So the sort of average microbiome profile for those 3 workflows is very similar. But the blue circles are bigger, meaning that there's just more variability in the microbiome profiles within those individual samples. Interestingly, the red and the yellow circles are almost stacked fully on top of each other. So both the sort of average profile and the variability in that profile is the same for the DNA and the fecal pools, which is really interesting. And that's in both ages of pigs. We can do some statistical testing on this and understand of the variability that we see in this plot, how much can sort of be explained or partitioned to various factors in our data set. And we did that, and we estimate that using something called an R-squared value, and age was the largest driver. So 25% of the variability in this plot is explained by, I'm going to put that in quotes, by age. Interestingly, sow ID accounted for about 18% of the variability. This is what I call the litter effect in swine production and microbiomes. The pigs they are raised together through weaning, and so they sort of converge into a very similar microbiome based on their litter membership. And then workflow, so the 4 different colors here, was about 5% of the variability. So a smaller amount, but it was statistically significant, and actually within like large microbiome-based cohort studies, 5% I say would be like a moderate effect size for workflow. So something we definitely need to pay attention to. So back to this question, our first question, is the overall composition different? It is when we compare individual versus pooled versus composite, but it's not when we just look at DNA pools versus fecal pools. Those were very similar, not statistically different from one another or biologically meaningfully different. So the second question, I just talked to you about overall composition, but another thing that people really like to look at in microbiome studies is diversity, alpha diversity. You know, people say, do you have a diverse gut microbiome? So we wanted to look at that as well and ask the same question. So to do this, we modeled first something called richness, which is basically the number of unique, let's say, unique microbes within the microbiome. And you know, some people think if you have more, that's better. That's not always the case, but a higher number indicates like a more unique bacteria within your gut, basically. And what I'm showing here is individual samples, fecal pools, DNA pools, and composite floor samples for the day one pigs. And you can see that, again, the composite floor samples, this is the average number of unique taxa within those samples, and then 95% confidence interval around that. It's just much higher, right, than the other 3 workflows. The DNA and the fecal pools, again, are basically on top of each other, indicating there's no real difference. And then the individual samples actually have a significantly lower richness than the fecal and DNA pools. So again, we see some differences based on workflow. And that was replicated in the day 20 pigs. There's other ways to look at alpha diversity as well, not just richness. People talk about Shannon's diversity a lot, and we see the same patterns, basically however we want to look at alpha diversity. So again, we have the same answer when we're looking at alpha diversity, which is that there are differences between individual, pooled, and composite, but when we're looking at the pools, the DNA versus the feces, they're the same. Okay, so individual samples are different. We know that, but I want to know why, because we know we're not going to do pen floor composite samples. They just aren't reflective. Do we have to do individual samples? Because that's where the money, that's where all the money goes, right? It's like all these 12,000 individual samples. I really like to be able to pool them at some level, but they are not the same as the pools. So I'm a little nervous, right? So how are they different? Let's dive into this a little bit. So this is an upset plot, and it's basically a Venn diagram, but I think easier to interpret. So here I have the 4 workflows again. And each of these bars represent the number of taxa, unique taxa, that were found in the various workflows. So if we look at this 1014 number, these 1014 unique taxa of bacteria, we found them in all 4 data sets. So no matter how we did the samples, we found those taxa. These 896 here, let's go down. They were only found in the composite floor samples. We did not find them when we looked in the other 3 workflows. So again, composite floor samples are not so useful. Okay, 648. This is the one that sort of worries me because these are the taxa that were only found in the individual samples, and we did not find them when we did the compositing or the pooling. So that's a lot of taxa, right? That's 648 taxa unique bacteria that we only find if we do individual processing of samples. So we wanted to understand them a little bit more in their distribution. What are these? I'm going to call them individual only ASVs, so individual only taxa. To do that, we did a scatter plot of all of the taxa found in the individual samples only. Okay, so each dot on this scatter plot is an individual taxa or ASV in the individual sample set. And the gray ones are the individual taxa that were found both in the individual samples and in the pooled and composite samples, okay? So those are the common ones across the workflows. The red dots are the 648 individual only ASVs, okay? So only found them in individual samples. Then on the y-axis, sorry, the x-axis, we put prevalence. So the percent of those individual samples that contained that taxon. And then on the y-axis, the relative abundance of each taxon. So up here, these would be like more abundant within each individual sample, and then down here would be less abundant. And what you can see is that the taxa or the ASVs found only in individual samples, they're like lined up right here. So they're very low prevalence. We only find them in like a couple of pigs within this 248 set of pigs, right? So they're very rare from a prevalence standpoint. And you can't see it as well because they're all stacked on top of each other, but most of them are clustered down here, meaning that they're also very low abundance within those samples. So these are the rare microbes that potentially, for some reason, they're just in some pigs and not others. They may be interesting, we don't know, but they are rare. We can say that. So with the conclusions from this that I presented today, I think we can say fecal pools are equivalent to DNA pools. Pen floor samples are not representative of individuals in the litter, so I would not recommend using them in pre-weaned pigs. This is different from the study I showed on like my first slide. And I think because that study was sampling post-weaned piglets. So there's reasons for that difference in result. And I think pooled samples can capture most of the diversity, but not some of those rare bacteria that you only get when you process individual samples. So with this, what's a poor swine microbiome researcher to do? I would say that you can do whatever you want, and I will not judge you because there's no clear answer here. But what we decided to do is for the large project, we ended up extracting individual samples and then pooling the DNA. So we created DNA pools and then we sequenced those pools. And we actually, Tara just submitted all of those 12,000 extracted samples that were then pooled to UMGC yesterday. So we will have results on that in a bit, but we did save a lot of money on library prep and sequencing. And yeah, we can always go back if we want to the individual samples because we have the individual DNA. So with that, thank you so much. And I'd love to take questions, suggestions, etc. Yes, Dr. Reed.
Let's get the mic here. Sorry for the folks on Zoom.
So you didn't mention what your sequencing depth is. Did you control for that in your individual versus pooled samples? So did you sequence at 4 times the coverage?
We did not. We did not. Yeah. So that's a great question. So one of the sort of question marks I had was this interaction between pooling and sequencing depth and how that would impact the microbiome results. What we did do is a rarefaction analysis. So we rarefied the data sets in silico after we got the data at different levels of sequencing depth and then reran the analysis. And those results are not in here. Generally speaking, the impact of sequencing depth is pretty equivalent across the different workflows. But I do think that, as you saw here, you lose those rare taxa more quickly within the individual samples because it's just a less even microbiome distribution in those. Mm-hmm.
So the difference in samples versus individual piglets versus pen. Obviously, the pen includes the sow as well.
Yes.
So could you individually sequence the sow and then do some sort of subtraction from pen samples?
Yeah. So, you mean in terms of the composite pen floor sample being so different? Yeah. So one of the reasons the pen floor composite I think is so different is because especially in the day-old piglets, they're just not producing feces really, right? So it's what we're sampling is the sow. And so subtracting microbiome data from other microbiome data is a really sort of slippery slope. People try to do that with contamination. They're like, these are contaminants, we're going to remove them, but it's really hard to tell what's a contaminant versus not. So I'd be like a little wary about doing that. I think more what I would maybe do is like sample the sow and say, this is the profile, this is how it overlaps or doesn't. And then people can make their own determinations as to like how much that's influencing the piglet samples. Yeah. Yep.
Any other questions? I'll ask one, Noel. I was really surprised by the the composite sample richness and how much is actually there compared to the swine samples. Do you think that has anything to do with just you're extracting it from a different sort of type of sample or like feces versus not feces, basically?
Yeah, I think actually what this has to do with is age of the pig that's being sampled from that's being represented in the pen floor sample. Because if you look at this, like the composite floor sample richness in the day 1 old piglets is around like less than 200. It's a little bit higher in the 20-day-old, but it's really the sow that's being represented in that sample. And we know from previous research that the microbiome of pigs, the richness and the diversity increases dramatically with age. So I think, again, you're seeing the effect of the sow. That's her poop on the floor. And so that's what you're getting, is that rich microbial profile of an older pig, basically. Yeah.
Any other questions? Do we have questions on Zoom? I am not moderating Zoom. Okay, well, with that, can we please thank Noelle one last time?
Thank you. Can you guys all hear me okay? Or is this recorded on the Zoom? Okay, great. Cool. So I'm going to introduce myself because all my grad students were either traveling or busy running flow cytometry today. So my name's Steve Friedenberg. I'm a faculty member in the clinical sciences department. Let's see, I'm a veterinarian. I went to vet school at Cornell. I did my internship and residency training in emergency and critical care medicine at Ohio State. And I decided I needed more torture after my residency and went on and got a PhD in genetics at North Carolina State. And I've been here since about 2017, and I'm really happy to talk to you guys about my research. It'll be very different from Noelle's presentation. This is a little bit of a culmination of maybe almost like 5 to 7 years worth of work. When you do work in the dog genetics world, a lot of times it's a lot more of a glacial pace. So we'll go through some of that today. Okay, so I study mostly autoimmune diseases in dogs as a geneticist, and I just wanted to give you a brief introduction of why I think dogs are a really good model for studying autoimmune diseases. So first of all, autoimmune diseases are very common diseases that we see clinically. I think when I decided to study autoimmune diseases, it was really because I saw a lot of them when they come to the emergency room, when I would take care of these patients in the ICU. And I just got really interested in these diseases because a lot of times patients get treated for autoimmune diseases, they'll die. Many of the diseases have high mortality. And then even if they do live, they're oftentimes subject to sort of lifelong immunosuppressive therapy, leading to secondary problems. And it's just kind of a mess. And so because they're such common problems, I really thought that these could use some addressing. They're also really similar to human diseases. Some of them, not all of them, that we see in dogs. And we'll sort of talk today about one disease that I study called Addison's disease. And I've for a long time thought of this as a really good model for Addison's disease in humans. As you'll see from some of the data that I'll present today, it actually might not be a great mimic for the same disease in humans, but it's sort of potentially worthy because they're similar diseases. They occur spontaneously. So, a lot of autoimmune disease research has been done studying mice and humans, especially if you're interested in human health, where a lot of the diseases that occur in mice are sort of induced models of disease. And so when we look and think about studying autoimmune diseases in dogs, they're in the same environment, they're subject to the same influences, they share a household with people. And so these diseases really occur naturally, and it's not necessarily an induced model. And I think one of the things I'll try to convince you of early on in this talk is that I think there's a lot of reasons why dogs offer a really advantageous genetic model for studying autoimmune diseases. And so whenever I sort of think about dogs as a genetic model, I like to throw up this figure here on the left, which actually comes from a paper by some of the kind of foremost people in dog genetics, Eleanor Carlson and Sharson Lindblad Toe from the Broad Institute in the University of Massachusetts at Amherst. And essentially, it sort of shows the formation of how dogs came to be. So, dogs and wolves at one point were all part of the same species. Maybe about 15 to 30,000 years ago, they diverged from wolves as humans started to domesticate wolves and sort of took under their wings animals that seemed to be more friendly, that were less fearful, et cetera. And then kind of for a really long period of time, sort of dogs existed as this sort of highly interbred population where there was tons of sort of genetic diversity across all of dogs. And then maybe I would say about 200, 300, 400 years ago, humans actually started to get heavily involved in what dogs look like and how they act and behave, right? So humans started breeding dogs to hunt. They started breeding dogs to look a certain way. They started breeding dogs to have certain types of hair coats, different sizes, all of those things. And these are the recent bottlenecks that have started sort of to occur and define sort of modern dog breeds. And what's happened when humans have had this intense artificial selection on how dogs should look or behave, it's created these things that we know and love of dog breeds, but these dog breeds have really, really limited genetic diversity within a breed. And because of this, certain diseases tend to become highly, highly overrepresented within dog breeds. So those of you who are veterinarians will sort of know this intuitively, but we tend to see a ton of autoimmune diseases that congregate within particular dog breeds. So for example, standard poodles, which is a dog breed that I study a lot of, they are sort of known for developing this disease called Addison's disease, which is a disorder of the adrenal glands, also a skin condition called sebaceous adenitis. Cocker spaniels are chock full of this disease called autoimmune hemolytic anemia and immune-mediated thrombocytopenia, which are autoimmune diseases that attack the red blood cells and platelets. Schnauzers, or miniature schnauzers rather, get diabetes and also another disease called hyperlipidemia. And German Shepherds are just chock full of autoimmune diseases, exocrine pancreatic efficiency, perianal fistulas, pannus, sort of a ton of different problems. But you can see all of these diseases are almost kind of like unique to the particular breeds of interest. And this really has to relate to the way in which dogs were formed and bred. And so when I think about genetic diseases and how we think of them, especially related to autoimmune diseases, when I sort of talk about how this breeding of an artificial selection has sort of impacted dog health, I sort of think of three different types of effects or diseases that we see within the dog world. And so the first kind of diseases are these Mendelian diseases, which are sort of single point mutations where you can ascribe one mutation leading to one disease. And the prevalence of, or sort of the association between the mutation and the disease tends to be really high. So the disease penetrance is the term we use. And most of these diseases form because you have these individuals like popular sires. So a good example would be, you have a disease that, let's say, a particular sire has, it doesn't manifest, and that this dog is beautiful, it's the right size, it has the right coat coloring, et cetera, et cetera. And so people will take this sire's semen and use it to breed, hundreds, if not thousands of females across a dog breed. Well, as it might turn out, when that sire becomes 5, he or she develops a particular genetic disease, and now that disease has been propagated all throughout the breed, right? So that's called a sire effect. You also have effects from sort of small number of founders. These create bottlenecks effects. And we have about, I think, 500 or so Mendelian diseases have been described in dogs. They're associated with single point variants. Now, when we think about autoimmune diseases, unfortunately, none of this applies because the vast, vast majority of autoimmune diseases that we see in dogs are polygenic diseases. And polygenic diseases means there's more than one gene that's involved in the development of the disease. There are also complex traits, which mean that the environment or environmental impact can also have an important effect as to whether or not a dog develops a disease. So even if you have all of the predisposing variants, you might not develop a disease unless you're actually exposed to some environmental effect. The other thing that makes life really challenging from a geneticist perspective is that many of the alleles or variants that come together to form complex traits are actually really common alleles or common variants that we see across the dog world. So kind of just to give you an example, there's a disease that's been where the genetics have been kind of thoroughly uncovered, and that's called dermatomyositis. And that's a skin condition that's seen in collies and Shelties. And we think that to get that disease, you need 3 different genetic variants to occur in a particular animal. Well, you could have the first variant might be common in Boston Terriers and standard poodles and German Shepherds, right? And the second variant might be common in Bull Mastiffs and English Bulldogs, and the third variant might be common in 3 other breeds. But those variants only come together and are common, all 3 of them, in Collies and Shelties. And so it becomes a lot harder to understand the genetics of these autoimmune diseases or these polygenic diseases because the variants are so common, and you have to start to understand how they come together in particular animals. The other thing that we are kind of learning from a lot of kind of current research in things that I do and things that other geneticists are doing is that many breeds, because of the way that we've sort of created them, have a fixed disease predisposition, meaning that all dogs can be predisposed to getting the disease. They have the same genetic background, but it's only when they have a particular environmental exposure that they'll actually get the disease. And understanding these fixed genetics effects requires different tools and techniques than we commonly use to sort through genetic diseases in animals, things like functional evidence that can be a lot harder to come by. So the research, so that's sort of just a little bit of a primer on dog genetics and creating dog breeds, and a little bit of a setup to the work that I do. And so my research really focuses on two different autoimmune diseases. The first one is a disease called Addison's disease, which is an autoimmune disorder that affects the adrenal cortex. It's certainly inherited in dogs and humans. It's highly overrepresented in certain breeds. And we'll talk a little bit more about Addison's disease in a second. And the other disease that I spend a lot of time thinking about is this disease called autoimmune hemolytic anemia. It's a disease where the body attacks its own red blood cells. And this disease is about 100-fold more common in certain spaniel breeds than other dog breeds. So potentially a good genetic model for both Addison's disease and autoimmune hemolytic anemia by focusing on certain breeds. Okay, and we're going to talk mostly about some of the work that I have ongoing in Addison's disease. So first I'll start and just explain a little bit about what Addison's disease is. And so Addison's disease is a disease of the adrenal cortex. The adrenal glands, if you're a human, live just on top of your kidneys. They're these triangular shaped organs. If you're a dog, they live just in front of your kidneys because dogs are kind of like humans, rotated 90 degrees. When you sort of look at the histology of the adrenal gland, it sort of helps us think a little bit about this disease a little bit more. So the adrenal gland is broadly divided into two parts. There's this outer layer called the adrenal cortex and the inner layer called the adrenal medulla. And in Addison's disease, it's only the adrenal cortex that's affected by the disease. If you were to double click and look detailed at the adrenal cortex itself is broken up into three different layers: the zona glomerulosa, fasciculata, and reticularis. And each of these layers of the adrenal gland secretes a different important hormone. So the outer layer of the adrenal cortex secretes a hormone called aldosterone, which is really important in salt and fluid balance in the body, as well as blood pressure. The middle layer secretes cortisol, which is a really important stress hormone in the body. And the inner layer, the zona reticularis, secretes certain androgens, which are really important in development and sexual function and all of these things. Interestingly, these androgens are less important in dogs than they are in people, at least the ones that are secreted by the adrenal gland. Okay, so what happens? In Addison's disease, the adrenal cortex gets destroyed or atrophied for some unknown reason. Without these hormones that are so vital to life, what do we actually see in clinical patients? So we start to see low sodium, high potassium, fluid losses, and changes in your blood pH. And these are all aldosterone-mediated effects that we see in the body. And you can sort of see why a patient with a disease like Addison's disease and an Addisonian crisis would present to the emergency room, right? Because they'll be severely dehydrated, they'll be in shock. High potassium can have really, really negative effects on the heart, causing bradycardia and ultimately cardiac arrest. So this is why many of these patients with Addison's disease will present to the ER where they come and see me, and hopefully we can get them better. And cortisol is really important stress hormone. And when patients don't have cortisol, enough cortisol floating around in their blood, we start to see hypoglycemia from impaired liver function, difficulty in maintaining the blood vessels and blood pressure in particular, absence of stress response, waxing and waning, vomiting and diarrhea. So a whole host of effects that we start to see in these patients clinically without these hormones. Okay. So let's talk now that we know a little bit about Addison's disease and what it is, a little bit about the genetics work that I've been doing to try to understand what's sort of the genesis of this disease. And so one of the things that I've said a couple of times is that certain autoimmune diseases are highly overrepresented in certain breeds. And when we look at different dog breeds, there have been some good studies out there, particularly from Sweden, where dogs are insured, and we can get good epidemiologic evidence of prevalence of diseases in certain breeds. And you can see that this standard poodles here have about a 17-fold relative risk of developing Addison's disease versus other dog breeds. This breed here is another breed that I study. Anyone know what breed this is here? This little guy? The Portuguese water dog. And so Portuguese water dogs, which are the nearest cousins of standard poodles, actually have a 29-fold increased risk of developing Addison's disease versus other dog breeds. This other dog breed here, which has a relatively high risk, is a bearded collie. And there are some other other dog breeds like Cocker Spaniels and Great Danes and English Springer Spaniels, and I'm not even sure what that is. I think it's a Nova Scotia duck-tolling retriever. They have slightly increased rates of developing Addison's disease, but not nearly as much as standard poodles and Portuguese water dogs. And so basically the main way in which we try to understand the genes or the markers that can separate affected from unaffected dogs is to do something called a genome-wide association study. I've just kind of thrown up here a quick cartoon to explain what a genome-wide association study is and how we do it. So this is just a random stretch of DNA many of you probably know that throughout the genome, we have these things that are called single nucleotide polymorphisms, which are variants throughout the genome that can be benign, they can have impact on the genome, but we sort of use these polymorphisms or variants as markers throughout the genome. And so, for example, this position here, you could see across a population of dogs, some dogs would have a G in this position, some dogs would have a C in this position. Here, some dogs would have an A, some would have a G. Here, some would have a C and some would have a T. And essentially what you do is you look at these different genotypes across a population of dogs and you try to find a marker. Right here, this is a marker in this case where all of these smiling standard poodles have a G in this position, and all of these sad standard poodles who have Addison's disease have a C in this position. And if you could find a marker that replicated like this across an entire genome, well, then maybe you'd have a marker that you could try to understand, well, what genes are in that region of the genome, and can we sort of find a mutation that might cause Addison's disease? So what I'm going to kind of show you on the next slide is really the culmination of about 5 or 7 years worth of work. And this is an actual Manhattan plot of looking at Addison's disease in standard poodles. And so what you can see is this plot is the result of something called low-pass whole genome sequencing. When you sort of look at changes in the way that genetic technology has changed over the years, we used to evaluate these markers on things called SNP chips, where you have in the dog world either 170,000 or 700,000 markers that you can genotype at the same time. In the past couple of years, we've started to move heavily away from using SNP chips and do something called low-pass whole genome sequencing. And what that is, it's where you take a dog genome, you sequence it to about a 2 to 3x coverage, and that's really, really low. Normally, when we sequence the genome of an animal, we sequence to about 20 or 30x coverage. But when you sequence at 2 to 3x coverage, it's much, much cheaper. So the cost differential is now about $60 to $90 for a low-pass genome versus $600 to $800 for a high-coverage genome. And what you can do is you can take these low-pass genomes and do something called genotype imputation, which is essentially a mathematical tool that takes the low-coverage genomes and in a haplotype, or for lack of a simpler term, a smart way, you can guess the genomes from the low-coverage data and impute them to the level of the high-coverage data. And so we did that in this case for about just under 100 dogs, 56 with Addison's and 37 without Addison's. After kind of pruning some of these SNPs for linked disequilibrium and also taking into account sex, we have about 1 million SNPs on this plot here. And you can see that there's a nice peak that comes up here on chromosome 17. And this is something on the right-hand side called a QQ plot, which lets us know, is our population well-balanced, and which it is by this measurement of something called lambda. And the other thing that we sort of look for, we look at these QQ plots, is we expect the p-values to kind of be the same until they're not, right? So these are the single nucleotide polymorphisms that are highly different between cases and controls. And this is kind of a really novel finding. So no one's ever been able to show a difference between affected and unaffected standard poodles before. And a lot of the reason why we've been able to do this is because we focus so heavily on getting our population so perfectly balanced in terms of relatedness of dog, in terms of sex, in terms of coat color, in terms of other things that can skew your genome-wide association results in dogs. And so the first question you ask yourself as a geneticist, okay, so it's nice to know that there's a peak on chromosome 17 at position 47.7 million. What are the genes that are kind of located right around the region where this peak is located? And one of the interesting things that we found in sort of looking in this region is we actually found that the only gene in this region is this gene called LRRTM4. And when you look at what this gene LRRTM4 does, it's actually been associated heavily with stress disorders and diseases like obsessive compulsive disorders in humans. And so none of the genes that we found in this initial GWAS seem to have anything to do with autoimmune diseases, which has actually been the dogma of what causes Addison's disease in standard poodles for years. And so kind of this is a little interesting, right? We found this GWAS, we have about 100 dogs, we have this kind of interesting peak here. We actually were able to, once you sort of do your initial GWAS, we were able to sort of expand our population from other samples we've collected to about 350 standard poodles. And when you expand this GWAS to a larger population, I'll just kind of focus your attention on the bottom pie charts here. You can see in controls, there's about a 50-50 differentiation between this risk allele of a T and the sort of normal allele of an A, whereas in cases, it's about 75-25. So even in a much larger population, genotyping this SNP still seems to have a really, really important effect. We came across another study after doing the sort of a separate genome-wide association study where people had actually looked at other dog traits like herding, predation, temperament, and trainability. And this other GWAS, interestingly, actually mapped the exact same gene and the exact same locus, but for totally different behaviors. So they looked at that, they found that same region to be associated with a lot of fear-related behaviors in dogs. And here we are mapping the same locus to dogs with Addison's disease. So what could be going on here? So one of the things that we've done as a follow-up, and this we actually have some new data just as of this morning. Fortunately, these sort of trends have held up. And so we asked a lot of patients or people who've enrolled in our studies to perform this survey called a CBARQ, which is a canine behavioral questionnaire, to try to understand whether dogs with Addison's versus dogs without Addison's have increased types of fear behaviors. And so when you look at these statistics, and these were kind of very small numbers when I put this slide together, so 9 and 22 dogs, we've now gotten up to about 30 dogs for each category. But a lot of these behaviors that are evaluated on the CBARQ, like excitability, leash pulling, dog-directed fear, there are some other ones that have kind of shown up, have all suggested that dogs with Addison's disease tend to be more stressed, more excitable, and really just have this hyperactivation potentially of their HPA axis, or the hypothalamic pituitary adrenal axis, that could be, and that some of these stress behaviors are highly, highly associated with Addison's disease in dogs. And so we're really in the process of trying to sort through exactly what this means and how many of these genes that we're finding or mapping could potentially be associated with what's historically been considered an autoimmune disease. I mentioned in kind of an early slide that another thing that we're learning is that fixed disease predisposition also seems, so having kind of the right genetic background also seems to have an important determinant as to whether or not you're going to develop an autoimmune disease. And so just to kind of show you this example, this is something called runs of homozygosity mapping. And so a run of homozygosity is a region where essentially all poodles are the same. And the x-axis shows you the chromosome, and the y-axis shows you how many dogs, how many standard poodles in our population actually have the run. And you can see we map these regions that are highly associated with body size, ear shape, furnishings, where it's these little things that kind of live on a dog's nose. We can actually map them easily by using runs of homozygosity. But there are these other runs of homozygosity where no one had really understood what's going on in these regions. And one of the things that we're finding when we look at these regions where all poodles are highly similar is we're finding other genes that are involved in the brain function and synapse function, sort of everything related from like potassium channels to G protein coupled receptors to things that modulate GABA receptors. So kind of a whole host of things. And everything we're mapping to, everything we're finding has to do with the brain. Kind of the last thing, which I'll just bring up before I finish, is that in addition to this genetics work, a year or so ago, I did a really big survey of dogs with Addison's disease, standard poodles and Portuguese water dogs, kind of a Qualtrics survey where we were trying to understand what are some of the environmental exposures that might be required for some of these genetic factors to be important. And so we sort of ultimately had a study population of about 5,000 dogs. And in doing this survey, we overlaid data from like water quality, air quality, soil quality data from the EPA, kind of wholly expecting to find something like, okay, you know, dogs with Addison's disease might be more exposed or like live in a Superfund site or something like that where they're exposed to environmental effects. Well, lo and behold, what we found is that humans have an outsized role again in the development of Addison's disease. So far and away, the biggest risk factor to developing Addison's disease that we found in this population is being neutered. So dogs that are neutered had a 6-fold increased rate of developing Addison's disease, and females that were spayed had a 2.5-fold increased risk of developing Addison's disease. So all of this speaks to the potential effect of losing some of these gonadal hormones influencing the adrenal gland and potentially could be having an outside effect on the way that the disease is developing. So that's kind of really all I'm gonna, that's the only kind of things I'm just gonna have to sort of sum up where we're going. This research is really trying to understand some of these MAP genes that have been associated with anxiety-related behaviors, synapse genes. How do these genes that have historically been thought of as brain genes, how are they affecting the adrenal glands? And really trying to understand, is Addison's disease in standard poodles at least truly an autoimmune disease? We also have kind of a parallel study going on in Portuguese water dogs, where we've done a preliminary GWAS that's actually been unremarkable in terms of finding any loci, but that's still a work in progress. And I think that's kind of all. I'll just stop there. I'll just kind of thank the folks who've been so helpful with me in this research, in particular the funders of all this. The Poodle Club of America Foundation has been funded a lot of this research, as well as the NIH, the AKC, the Portuguese Water Dog Foundation, all of my lab members, and in particular, Leanne Clark, who's a great collaborator of mine at the University of Georgia, has been kind of instrumental in this work. And that's all I'll say. So thank you, guys.
Any questions? Got one over here.
I'm curious, how has like advancements in genetics impacted dog breeding generally?
It's a good question. You know, I think one of the ways in which advances in genetics has affected dog breeding is there's a lot of commercial services now available where you can sort of have your dog genotyped in one fell swoop for essentially all of the mutations that have been ever found in any dog breed. So I think that certainly affected breeders' breeding behaviors. You know, there are some kind of controversies with a lot of these things. So for example, let's say you have a poodle, but there's been a mutation discovered with a heart disease in Doberman pinschers, you'll still find out the result of that test in your poodle, and it kind of may or may not be important to a poodle because it's on like a totally different genetic background. But that's certainly been one way in which it's helped breeding. Certainly, if you have a mutation that's been discovered in a particular breed, breeders will start to breed away from that mutation. That can have outsized consequences because depending upon how prevalent that mutation is, you could kind of further restrict the diversity of your limited breeding pool. So you sort of have to take all these, you have to think about the effects that your breeding decisions are going to make. And one other thing that we can also do like nowadays, if people want to sequence the entire genome of their animal to look for rare mutations, I think that's kind of another thing that we can do or we can offer to owners. Question from the back. Yeah, hey,
yeah, Steve, yeah. Great talk, very interesting. So I have two related questions. One is serious, the other one, it's up to you all.
Sure.
The question is about, you know, this potential link between the Addison's disease and the, let's say, anxiety in these dogs, and your intent to maybe use this as a model for humans, knowing the prevalence of anxiety in people, any relationship, that genetic relationship that you know or that you've seen with the Addison's disease and anxiety in people. And then that's the serious question. And the other one is, you know, we joke that the owners look like their dogs or vice versa, right? Yeah. Anything in that respect?
So on the last question, you know, sometimes you feel like I go to a lot of dog shows, and sometimes I feel like I see a standard poodle that looks a little bit like its owner, or the owners will have the curly hair, and then they'll give like the poodles like a pink tail or like blue feet, and the owners will try to dye their hair the same way, but nothing genetic related that I could think of. So in terms of the human situation, so it's actually really interesting. In humans, there have been studies where about 90% of humans that develop Addison's disease will develop autoantibodies against a protein called 21-hydroxylase, which is an important protein in the adrenal gland steroidogenesis. And so I think we know, and there's also sort of documented associations between particular human leukocyte antigen alleles and Addison's disease. And so it's pretty well documented that Addison's disease is an autoimmune disease in people. We've looked for these autoantibodies in dogs and have never been able to find them, and other people have as well, have been unable to find them as well. Other people have looked but have not been able to find them. And maybe it speaks to sort of we're not doing a good job at trying to find them, but I think it also might speak to the fact that Addison's disease is actually not an autoimmune disease in dogs, and you have something where a totally different mechanism of action actually leads to sort of the same output. And there's actually other nits in the dog world in terms of like subclasses of Addison's disease that I didn't really have time to get to that don't exist in people that actually might speak to atypical. If you're a clinician, like atypical Addison's disease, right? That doesn't exist in people, and so that might speak to actually a slightly different pathogenesis between dogs and people. So, but it actually, even though it might not be a great model for human Addison's disease, I don't know if anyone's ever heard of this quasi-scientific term called adrenal fatigue. This was like something that was popularized by like Suzanne Summers when she like wrote a book a couple years ago. When you sort of talk to most endocrinologists, they completely like poo-poo that idea as if it's sort of not something that really exists. But maybe this is a model for something like that really no one's able to kind of suss out those effects between the brain and the adrenal gland. And maybe this will kind of turn into something that kind of helps us understand the relationship between the brain and the adrenal glands and the gonads like a lot better. So sorry, that was a long-winded answer.
Okay. We're about wrapped up here for time. So can we thank Steve one more time? And then I just want to plug our next seminar, which is going to be the second Wednesday of June. So I'd like to see you all there. And then in July, we aren't going to have a RIDE seminar because it's going to be our RIDE summit. So it's going to be the presenting research from across the college at the RIDE summit. So look for details on that. And if you've seen or you all have been to this seminar series, please give us feedback. You can find we have a short survey to take, which we're going to try to get the QR code up, but we send the QR code also with all the invitations, and you can find it on the research office website. So thank you all for being here. I really appreciate it, and see you all next time.